Hello children, I hope you are doing well. Last class we saw about lymphoid organs and today we'll see about AIDS. Okay, I think you're all you'll be familiar with AIDS and the causative agent, how it spreads, how to control and prevent AIDS. We'll see all these things today. AIDS. What is the full form of AIDS? Acquired immunodeficiency syndrome. See, acquired immunodeficiency syndrome. For all these, everything has got a meaning. See, it means deficiency of immune system acquired during the lifetime of an individual. The individual is acquiring, the individual is acquiring a deficiency in his immune system. It is not a congenital disease. It is not inherited from the parents. He is acquiring during his lifetime. Okay, deficiency of the immune system. Okay, syndrome. Syndrome means a group of symptoms. See, when a patient is having AIDS, he will show other symptoms, other diseases also. So that is only, it's a syndrome. It is a syndrome, acquired immunodeficiency syndrome. Syndrome means a group of symptoms. And this was first reported in 1981 and it has spread all over the world. Okay, it has spread all over the world. This is what it is. AIDS is caused by human immunodeficiency virus, a virus, HIV virus. Okay, and we'll see again one more picture. This is actually it's caused by HIV virus, that is human immunovirus. This virus it comes under the category retroviruses. Actually, this HIV virus belong to a group known as the retroviruses. Actually, this is the structure of a retrovirus. It will be HIV virus also similar to this only. Okay, same thing. This actually HIV comes under group retroviruses. The same structure it will be having. Here we can see the envelope, the covering for the virus. It has got proteins and lipids as an envelope, and inside you have the viral enzymes and the viral RNA. It will be having an enzyme known as the reverse transcriptase, and this enzymes plays a vital role in converting the viral RNA into viral DNA when it enters the host body. Okay, inside it will be having the core. Okay, inside that protein core, outer layer envelope made up of protein and lipids. Similar, actually the same thing. Next we'll see the picture of HIV. This is what the HIV virus. I said it is also here. It has got the enzyme. Okay, enzyme. This surrounding the proteins with the lipid, and this enzyme converts the RNA into DNA. This is the picture of a HIV virus enclosing a RNA genome. Okay, now we'll see transmission of HIV, how the transmission of HIV infection occurs. Okay, how it's the one person to another person, how it spreads. Transmission of HIV infection occurs by sexual contact with the infected person. If the person has got HIV and that person having a sexual contact with him also gets HIV. Then transfusion of contaminated blood and blood products by mistake. Very poor monitoring. If the blood and the blood products infected not only with HIV but other diseases also, the patient might get this. And sharing infected needles. Okay, blood, I said first blood and the blood products, and sharing infected needles as in the case of intravenous drug abusers. Okay, they have drug addicts and all, they have the habit of sharing needles. If one person has got HIV, if the other person shares, he will get it, HIV. okay, and then infected mother to a child through placenta. See, here the child gets HIV through the mother during the time of pregnancy in the placenta, through the placenta. This is also by which transmission of HIV. This is also by which HIV get transmitted to the baby from the mother. Okay, so this is what transmission of HIV infect occurs by. I've told you four things. 1, 2, 3, 4. So all these ways the infection can occur. Okay, then we'll see people who are getting at a high risk of people who are at a high risk of getting this infection. It includes individuals who have multiple sexual partners. Okay, so they are likely to get, and drug addicts who take drugs intravenously. Their chances are there for them, and individuals who requires repeated blood transfusion. Some of the patient they require repeated blood transfusion. That is in the case of anemia, if they are having anemia or sickle cell disease, hemophilia or cancer. Those patients they need a repeated blood transfusion. If it is not done properly, they might get this HIV infection. And children born to HIV infected mother. So these are the by which the people can get the infection. These are the ways. Okay, and HIV, you must know that HIV or the AIDS actually here diagram is given which we'll see later on about this one, how it goes inside the thing. HIV is not spread mere by touch or physical contact. That must be aware. It spreads only through the body fluids, including blood. It's also a fluid. It spreads only through body fluids. People should be aware of this. It is not spread by mere touch or physical contact. So it is hence important for the physical and psychological well-being for the HIV patient, infected person. Or not, we must not isolate. Okay, they are not isolated from family and society. We have to take good care of them. Okay, and this one more thing is there is always a time lag between the infection and the appearance of the AIDS symptoms. That is, people might get the disease, but they will not show the symptom. Okay, after incubation, that is what known as incubation period. It varies from one disease to another. Okay, for this particularly HIV, this period may vary from a few months to many years. Usually 5 to 10 years. The patient will be having HIV, but they will not show any symptoms. It will take longer time. So this 5 to 10 years means that time it will come to an end. It will be very critical. So what is that early checkup? They can go in for a checkup. Nowadays you have a ELISA test, which is a very prompt test for this HIV. So they can go in for a test because incubation period is too much. So, and this picture now we'll see. This is what the replication of retroviruses. See, retroviruses, I showed the picture and all. Viral RNA, it will be having the viral RNA core. The virus infects the normal cell. You can see the outer protein coat, how the virus goes inside animal cell or human cell. We have the plasma membrane. Cytoplasm is there. The viral RNA is introduced into the host cell. What happens here? Immediately it is introduced into the host cell. The viral DNA is produced by reverse transcription. This is what the enzyme I showed, which will be seen in the virus. Okay, so viral DNA. So the RNA genome will be converted into DNA. Viral DNA is produced by the reverse transcriptase, and this viral DNA will be just incorporates into our host genome, our DNA. It's a nucleus DNA. A new viral DNA is produced once it goes inside. New viral DNA will be produced by the infected cell. Okay, you can see this infected cell, and new viruses or this once the cell get infected again and again, this one. Okay, new viral it's produced by the infected cell, and new viruses are produced, more viruses are produced. Okay, you can see once the viral RNA encroaches into our genome DNA, this is nucleus. Again, new viral DNA is produced by the infected cell. Again, new viral, and again this one. New viruses are produced here. More viruses are produced, and these new viruses can infect other cells. Okay, infected cell can produce, and again these viruses will be infected fresh cells, and again it will all the cells become virus infected cell. Once the infected cell it can survive while viruses are being replicating, making exact copies and released, this can survive. Okay, like this the infection keeps on going in our body, and our cells will be converted into viral particles. Okay, this I will explain now as it is given in your book. We'll see this. Okay, I'll give the explanation. The same picture, but a little more in detail. See what happens here after getting into the body of the person. What happens? HIV virus enters into macrophages. See, this macrophages we have already seen when we studied about the immune system. It's a type of WBC of the immune system. It's a type of WBC. Macrophages means engulfing and destroying any foreign antigen or any foreign antigen or a pathogen, and it will be very stationary form in the tissue. Okay, when any antigen enters, it will try to encounter and destroy the antigen. Okay, so what happens is virus straight away enters into the macrophages, where the viral RNA genome of the virus replicates, making exact copy to form, or it gets converted to form viral DNA with the help of the enzyme reverse transcriptase. So this is the first thing that takes place inside our body. What happens here? The virus enters the macrophages, where what happened? The RNA genome, viral RNA genome gets converted into viral DNA because of the presence of the enzymes. That this viral DNA gets incorporated into the host cell DNA. That is what I showed in the picture. Once it gets converted into viral DNA, it's getting incorporated into the host cell DNA and directs the infected cell to produce viral particles. Once it gets incorporated into the host cell DNA, our cell becomes infected. So it directs the infected that cell to produce virus particles. The macrophages continue to produce virus. Okay, and in this way they will be producing more and more virus. It'll act like a HIV factory. Then what happens? HIV enters the T lymphocytes. Already this also you have seen in the immune system. Replicates, makes exact copies, and produce progeny viruses, young viruses. The progeny viruses released in the blood attack other T helper cells. This is repeated in all our T helper cell. It gets converted into viral particles, and there will be a rapid decrease in the number of the T helper cells, T lymphocytes, in the body of the infected person. That is what they become immune deficiency in the immune system. During this period, okay, the person suffers from attack of fever or diarrhea, weight loss. Okay, weight loss. And this macrophages, these are the cells macrophage. I said no macrophages. First, it will infect the macrophages. Okay. And all these, they will get frequent fever, diarrhea, weight loss, everything, because they will be having a very weak immune system or completely loss. Okay, loss of due to the decrease in the number of T lymphocytes, a person gets other infection. That's what I said first. It's syndrome, other infections, so other bacterial, fungal, viral infection, and sometimes even parasites like toxoplasma. That's nothing but like malarial. We saw spore forming protozoan. So the patients become immunodeficient. So that is a critical stage. It's very early stage. It's curable. That's all. So the diagnostic test for AIDS is ELISA. ELISA is nothing but enzyme linked immunosorbent assay. See, this ELISA test is nothing but they take a patient's blood sample for antibodies. Sometimes they take the oral fluid also, and they test the blood and measure the antibodies in the blood. So sometimes I already said whenever there is an infection only, our antibodies in our blood will be in a high level. So they will just, antibodies are specific for a particular antigen, specific antibodies. So they will measure the antibodies in the blood, and there will be more antibodies produced in response to the harmful substance called the antigen. So they will find out whether you have HIV or not. This is a very good test. Early detection is good, and treatment is nothing but antiretroviral drugs are there, but only partially effective. Early stages it is good, they can prolong the life of the patient, but only you can prolong the life. Okay, that's all. So prevention of AIDS, we know no cure, permanent cure is not there, so prevention is the best option. Okay, so that is very important. HIV infection spreads due to conscious behavioral patterns. That is what. So sometimes it happens in hospitals and all during a blood transfusion time. If it's infected blood, patients might get infection, or newborn from mother may take place. That is all because of poor monitoring. Okay. So the only thing is maybe because of ignorance. All these happens because of our ignorance. Don't die of ignorance. See, in our country we have organizations like National AIDS Control Organization. They are doing a number of programs to control AIDS, and other non-governmental organizations also in our country. They are doing a number of lot of programs to educate people about AIDS, and World Health Organizations have started a number of programs to prevent the spreading of this HIV infection. Okay, what and all they are doing is they are making blood safe from HIV, ensuring the use of only disposable needles and syringes in all public and private hospitals and clinic. That is very good. Disposable, we need not use the same thing. Okay, once used, that's all, you have to throw it. And free distribution of condoms, and they are controlling drug abuse, advocating safe sex, and regular checkup for HIV in susceptible populations. These are some steps taken by World Health Organization. Okay, and the non-governmental having AIDS should not be hidden. Okay, that should not be hidden. They need help and sympathy. Okay, and it is an ailment or a malady. It's a malady in the sense it's an ailment or a disease that can only be tackled by the society. and by medical fraternity. Acting together, they can control and to prevent the spread of the disease. Both must together act, and they have to control the spread of the disease. And the AIDS patient need help and sympathy, so we have to show uh, our sympathy, and we have to be very loving and caring. Okay, that is very, very important. Thank you, children. Next class we'll see about AIDS. I'm sorry, next class we'll see about cancer.